Very good morning, dear students. So, welcome back to 2023 science examination preparation. So, I hope you all know that we have started learning the heredity and evolution lesson for the final examination. Okay, this video is all about final science examination preparation related. So, welcome back to each and everyone who are present in today's live. So, ಇವತ್ತಿನ class ಅಲ್ಲಿ I am going to discuss about the heredity and evolution, my dear students. So, you know that when we look at the model question paper, so we have heredity and evolution lesson question for 5 mark. So, 5 mark ಆಗಿರಬಹುದು 6 mark ಆಗಿರಬಹುದು ಈ lesson ಇಂದ questions ಅನ್ನ ಕೇಳ್ತಾರೆ. So, heredity and evolution ನ every bit of concept ನಾನು ನಿಮ್ಮ ಜೊತೆ ಡಿಸ್ಕಸ್ ಮಾಡ್ತಾ ಇದ್ದೀನಿ. So, we have started discussing the concepts from the lesson heredity and evolution. So, in today's class I am going to continue further. So, yesterday was the first session. So, yaradru previous session videos na nodilla andre do watch the previous session videos. So, you will know the basics of heredity and evolution, and it is easy for everybody if you know the basics to answer for every question which will be asked in the final examination. Okay. So, if you are new to my channel, do subscribe the channel and also do share the videos with your friends. So, nimge see previous class alli ನಾನು ನಿಮಗೆ ಕೆಲವೊಂದು ಕಾನ್ಸೆಪ್ಟ್ನ ಹೇಳ್ಕೊಟ್ಟೆ. ಬೇಸಿಕ್ಸ್ ವಾಟ್ ಈಸ್ ಹೆರಿಡಿಟಿ ಅನ್ನೋ ಕಾನ್ಸೆಪ್ಟ್ ಹೇಳ್ಕೊಟ್ಟೆ. ಆ್ಯಂಡ್ ದೆನ್ ವಿ ಹ್ಯಾವ್ ಡಿಸ್ಕಸ್ಡ್ ಅಬೌಟ್ ವಾಟ್ ಈಸ್ ಮೀನ್ ಬೈ ಡಾಮಿನೆಂಟ್, ವಾಟ್ ಈಸ್ ಮೀನ್ ಬೈ ರಿಸೆಸಿವ್, ಮೆಂಡಲ್ಸ್ ಮೋನೋಹೈಬ್ರಿಡ್ ಚೆಕ್ಕರ್ ಬೋರ್ಡ್, ಪಿ ಪ್ಲಾಂಟ್ಸ್ನೇ ಯಾಕೆ ಮೆಂಡಲ್ ಚೂಸ್ ಮಾಡಿದ್ರು. ಓಕೆ, ಆಲ್ ದೋಸ್ ಥಿಂಗ್ಸ್ ವಿ ಹ್ಯಾವ್ ಅಂಡರ್ಸ್ಟುಡ್. ಬಟ್ ಇನ್ ಟುಡೇಸ್ ಕ್ಲಾಸ್ ವಿ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ಫರ್ದರ್ ಫ್ಯೂ ಮೋರ್ ಬೇಸಿಕ್ ಕಾನ್ಸೆಪ್ಟ್ಸ್ ಆಫ್ ಹೆರಿಡಿಟಿ ಆ್ಯಂಡ್ ಎವಲ್ಯೂಷನ್. ಸೊ ಅದರಲ್ಲಿ ಫಸ್ಟ್ ಕಾನ್ಸೆಪ್ಟ್, ವಾಟ್ ಈಸ್ ಮೋನೋಹೈಬ್ರಿಡ್ ಕ್ರಾಸ್. What is mono? Mono is nothing but single, children. Okay, mono is nothing but single. Okay, so cross is nothing but which is this is the one thing which is taking place or taking part between two parents or two offsprings in the next generation. Okay, how can we define monohybrid cross? See, it is a cross between two pea plants. Not only pea plants, you can take any kind of plants, but now illi pea plants related agi discuss ಮಾಡ್ತಿರೋದ್ರಿಂದ I have mentioned two pea plants. So, and these two pea plants will differ in only one character. This is very important. See, when I take capital T and small t, so this capital T and small t is nothing but they both are related to height. So, here height is the one character. Height is the one character which we have taken. Okay. So, we are differing by only one character, and these two are the this is tall and dwarf is the contrasting characters. Okay, if you take the white flower and also let us take the pink flower, so this is also one character, but they are the contrasting characters. Okay, and you must know what is the phenotypic ratio. Phenotypic ratio ಏನಾಗಿರುತ್ತೆ? What is the phenotypic ratio of the Mendel's monohybrid cross experiment? Always the phenotypic ratio is 3:1. So previous class ಅಲ್ಲಿ ನಾನು ನಿಮಗೆ ಹೇಳಿದ್ದೀನಿ ಯಾಕೆ 3:1. So when we cross the tall pea plant with the dwarf pea plant, when we cross the tall pea plant versus dwarf pea plant, in the F1 generation we got all tall pea plants, correct, on cross pollinating, and when we self pollinate these pair of alleles, we got something like this, that is capital T, capital T, small t, two set of capital T, small t we got, and we got small t, small t. So we have observed that totally we have 3 tall pea plants and 1 dwarf pea plants. So based on this we wrote the phenotypic ratio. Based on this we are writing the phenotypic ratio. So all the things which I am explaining is definitely going to help you, my dear students. You have to follow the concept ಬಗ್ಗೆ ನಿಮಗೆ ಗೊತ್ತಿರಬೇಕು. Okay. So next question is, what is dihybrid cross? What is meant by dihybrid cross? See, it is a cross between two plants. Dihybrid cross is a cross between the two plants. But now we are differing with the two specific character here. Earlier ಏನು ಮಾಡಿದ್ವಿ, ಒಂದೇ ಕ್ಯಾರೆಕ್ಟರ್ ಇತ್ತು, ಬಟ್ ಇಲ್ಲಿ 2 ಕ್ಯಾರೆಕ್ಟರ್ ಇದೆ. Let us now take tall red flower, tall plants with red flower, and let us take dwarf plant with the white flower. So you can take any character, but we have 2 character, tall and dwarf ಅನ್ನೋದು ಒಂದು character, that is the height, and ಇಲ್ಲಿ color, so white and the pink ಅನ್ನೋದು another color. Okay, so I can denote by any letters. So I can also take round and wrinkled. ರೌಂಡ್ ಅಂಡ್ ರಿಂಕಲ್ಡ್ ಆಗಿ ಕೊಟ್ಟಿದ್ರೆ ಕ್ಯಾಪಿಟಲ್ ಆರ್ ಸ್ಮಾಲ್ ಆರ್ ತಗೊಳ್ಳಿ. And if you don't want to put this letter for white, you can take WW. So white and its contrasting character is pink. Okay, or you can take white flower with the yellow flower. You can choose anything, children. But mainly what we are focusing, focus ಮಾಡ್ತಾ ಇದೀವಿ ನಾವು ಇಲ್ಲಿ ಅಂದ್ರೆ, so ಇಲ್ಲಿ two characters ಇದೆ. There are two characters. Okay, two contrasting characters are there. So when we cross between two contrasting character, then it is called as the dihybrid cross. ಎಕ್ಸಾಮ್ ಅಲ್ಲಿ ನಿಮಗೆ ಕ್ವೆಶನ್ಸ್ ನ ಕೊಟ್ಟಾಗ ಒಂದು ಇದೆಯಾ ಕ್ಯಾರೆಕ್ಟರು, ಎರಡು ಕ್ಯಾರೆಕ್ಟರ್ ಇದೆಯಾ ಅಂತ ನೀವು ಐಡೆಂಟಿಫೈ ಮಾಡಬೇಕು. ಸೊ ಆಲ್ವೇಸ್ ದ ಫಿನೋಟೈಪಿಕ್ ರೇಷಿಯೋ ಆಫ್ ದ ಡೈಹೈಬ್ರಿಡ್ ಕ್ರಾಸ್ ಈಸ್ 9:3:3:1. ಯಾಕೆ ಅಂತ ನಾನು ಇವತ್ತು ನಿಮಗೆ ಡೈಹೈಬ್ರಿಡ್ ಚಕ್ಕರ್ ಬರೆಯೋದು ಅಂದ್ರೆ ಚಕ್ಕರ್ ಬೋರ್ಡ್ ಬರೆಯೋದನ್ನ ಹೇಳ್ಕೊಡ್ತೀನಿ. ಸೊ ಯು ವಿಲ್ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್. ಓಕೆ, ಯು ವಿಲ್ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ದ ಫಿನೋಟೈಪಿಕ್ ರೇಷಿಯೋ ಆಫ್ ದ ಡೈಹೈಬ್ರಿಡ್ ಕ್ರಾಸ್. ಓಕೆ, ಸೊ ವೆನ್ ಯು start writing the second generation, definitely you will come to know what is the phenotypic ratio of the dihybrid cross. So you must know the difference between monohybrid and dihybrid. Let us now go for next concept based question on heredity and evolution lesson. What is F1 generation and what is F2 generation? See, we always get offspring in one generation, the first generation, we call it as F1 generation. We will also get offspring in the F2 generation. Okay, so we get more offsprings in F2 generation. So now what is this F1, what is this F2? See, F1 generation is nothing but the first generation. F1 generation andre enu? F1 generation andre first generation anta artha. So first generation offsprings which was produced by the first set of parent. ಈಗ ಫಸ್ಟ್ ಸೆಟ್ ಆಫ್ ಪೇರೆಂಟ್ ಯಾರಿದ್ರು, ಈಗ ಟಾಲ್ ಅಂಡ್ ಡ್ವಾರ್ಫ್ ಅನ್ನೋರು ಫಸ್ಟ್ ಸೆಟ್ ಆಫ್ ಪೇರೆಂಟ್. ಫಸ್ಟ್ ಸೆಟ್ ಆಫ್ ಪೇರೆಂಟ್ ಇಂದ ಪ್ರೊಡ್ಯೂಸ್ ಆಗಿರೋರೆ F1 ಜನರೇಷನ್ ಆಫ್ಸ್ಪ್ರಿಂಗ್ಸ್. ಈ ಫಸ್ಟ್ ಸೆಟ್ ಆಫ್ಸ್ಪ್ರಿಂಗ್ಸ್ ಏನಿದೆ, ಈ ಆಫ್ಸ್ಪ್ರಿಂಗ್ಸ್ ಅನ್ನ ನಾವು ಸೆಲ್ಫ್ ಪಾಲಿನೇಟ್ ಮಾಡಿ ಅಗೇನ್ ವಿ ಗೆಟ್ ಫ್ಯೂ ಮೋರ್ ಆಫ್ಸ್ಪ್ರಿಂಗ್ಸ್. ಸೊ ದ ನೆಕ್ಸ್ಟ್ ಸೆಟ್ ಆಫ್ ದಟ್ ಈಸ್ F2 ಜನರೇಷನ್ ಇಸ್ ದ ಸೆಕೆಂಡ್ ಫಿಲಿಯಲ್ ಜನರೇಷನ್. ಓಕೆ, ಸೊ ದಿ ಸೆಕೆಂಡ್ ಫಿಲಿಯಲ್ ಆಫ್ಸ್ಪ್ರಿಂಗ್ಸ್ ಆರ್ ದ ರಿಸಲ್ಟ್ ಆಫ್ ದ ಪೇರೆಂಟ್ಸ್ ಆಫ್ F1 ಜನರೇಷನ್. ಅಂದ್ರೆ F1 ಜನರೇಷನ್ ಅಲ್ಲಿ ಬಂದಂತ ಆಫ್ಸ್ಪ್ರಿಂಗ್ಸ್ ಏನಾಗಿರ್ತಾರೆ, F2 ಜನರೇಷನ್ಗೆ ಪೇರೆಂಟ್ಸ್ ಆಗಿರ್ತಾರೆ. ಓಕೆ, ಸೊ ದಿಸ್ ಇಸ್ F2 ಜನರೇಷನ್. F2 generation is nothing but second filial generation. F2 generation ಅಂದ್ರೂ ಒಂದೇ, second filial generation ಅಂದ್ರೂ ಒಂದೇ. ಓಕೆ, F2 generation ಅಂದ್ರೂ ಒಂದೇ, second filial generation ಅಂದ್ರೂ ಕೂಡ ಒಂದೇ. Both are same. Okay, so you must know F1 generation is nothing but first generation offsprings which are produced by the first set of parents. F2 generation is also called as ಸೆಕೆಂಡ್ ಫಿಲಿಯಲ್ ಜನರೇಷನ್. ಈಗ F1 ಅನ್ನು ಕೂಡ ನಾವು ಫಸ್ಟ್ ಫಿಲಿಯಲ್ ಅಂತ ಹೇಳ್ಬೋದು. ದಿಸ್ ಇಸ್ ದ ಫಸ್ಟ್ ಫಿಲಿಯಲ್. F1 is the first filial, F2 is the second filial. So you must know F1 is same as a first filial. Okay, F2 generation is nothing but second filial generation which are produced as a result of the interbreeding between the which parent, the F1 generation offsprings. ಓಕೆ, F1 ಜನರೇಷನ್ ಆಫ್ಸ್ಪ್ರಿಂಗ್ಸ್ ನೇ ನಾವು F2 ಜನರೇಷನ್ ನ ಪೇರೆಂಟ್ಸ್ ಅಂತ ಹೇಳಿ ಕರಿತೀವಿ. ಸೊ ಇಟ್ಸ್ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್. ಸೊ ಮೂವಿಂಗ್ ಟು ನೆಕ್ಸ್ಟ್ ಕಾನ್ಸೆಪ್ಟ್. ಸೊ ದ ನೆಕ್ಸ್ಟ್ ಕಾನ್ಸೆಪ್ಟ್ ವಿಚ್ ಐ ಆಮ್ ಟೀಚಿಂಗ್ ಹಿಯರ್ ಇಸ್ ಎಕ್ಸ್ಪ್ಲೈನ್ ದ ಮೆಂಡಲ್ಸ್ ಡೈಹೈಬ್ರಿಡ್ ಕ್ರಾಸ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್. ಮೆಂಡಲ್ನ ಡೈಹೈಬ್ರಿಡ್ ಕ್ರಾಸ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ನ ಎಕ್ಸ್ಪ್ಲೈನ್ ಮಾಡಿ. ಸೊ ಐದರ್ ದೇ ಆಸ್ಕ್ ಇನ್ ದ ಎಕ್ಸಾಮಿನೇಷನ್ ಮೊನೋಹೈಬ್ರಿಡ್ ಆರ್ ಡೈಹೈಬ್ರಿಡ್. ಸೊ ರೀಸೆಂಟ್ ಕ್ವಶನ್ ಅಲ್ಲಿ ನಿಮಗೆ ಜೆನೆಟಿಕ್ ಮೇಕಪ್ ಬರೀರಿ ಅಂತ ಕೇಳಿದ್ರು. Most of the students don't know what is meant by genetic makeup. Genetic makeup andre enu is nothing but genetic makeup is nothing but the genotype of that particular genotype of that particular cross. Genetic makeup is nothing but the genotype. Remember, okay? Yes. Dihybrid cross experimentally, what do you learn? So you learn about the crossing between two parents. So the first parent here which we are taking is the round. So here when Mendel is performing this set of experiment, he has chosen the two contrasting characters. The first contrasting character was the shape. Shape was the one contrasting character he has chosen, and the color was the another contrasting character which he has chosen. So what is the shape he has chosen? He has chosen round. So and also the wrinkled. He has chosen round and wrinkled. Later he has chosen the yellow and also the green. Yellow and the green. So these are the characters which are being chosen by the Mendel. So round is the shape of the seed, and wrinkled is the shape of the seed. Yellow and green is the color of the seed. So he has taken two character with the contrasting traits. Okay, so that is why it is called as dihybrid. Di is nothing but two. So what he did, ಏನು ಮಾಡ್ದ ಅವನು? ಸೊ ಮೆಂಡಲ್ ಏನು ಮಾಡ್ದ ಅಂತಂದರೆ ಈ ಎರಡನ್ನೂ ತಗೊಂಡು ಕ್ರಾಸ್ ಪಾಲಿನೇಟ್ ಮಾಡ್ದ. He has cross pollinated the round yellow seed with the wrinkled green seed. So cross pollinate ಮಾಡಿದಾಗ he has obtained something like this in his F1 generation. What did he got? He got something like this. So he has received one trait from the round, that is one allele ಅಂತ ಹೇಳ್ತೀವಿ ನಾವು, ಒಂದು ಲೆಟರ್ನ ಒಂದು allele ಅಂತ ಹೇಳ್ತೀವಿ. So one allele from round and one allele from wrinkled, one from yellow and one from green. So he has got something like this. So what is this? So always we have to look for the traits which are getting expressed in the F1 generation are dominant. So capital letter ಅನ್ನೋದು ಏನಿರುತ್ತೆ, capital letter ಏನಿರುತ್ತೆ round, and capital letter ಇಲ್ಲಿ ಏನಿದೆ yellow. Okay, capital letters are expressing their traits. ಯಾಕೆ? Because these letters are dominated. They are dominated. So the offspring which we got in the F1 generation, all the seeds are round yellow in shape. The offsprings which we got in the F1 generation, all the seeds are round yellow in shape. Round seeds with yellow. So next, to perform next set of experiment, that is the F2 generations experiment, what he did was, he has taken this offspring, F1 generation offspring as a parent. ಇವನು ಏನು ಮಾಡ್ತಾನೆ ಅಂದರೆ ಸೆಲ್ಫ್ ಪಾಲಿನೇಟ್ ಮಾಡ್ತಾನೆ. ಫಸ್ಟ್ ಕ್ರಾಸ್ ಪಾಲಿನೇಟ್ ಮಾಡ್ದ. ಸೊ ನೆಕ್ಸ್ಟ್ ಏನು ಮಾಡ್ತಾ ಇದ್ದಾನೆ, ಸೆಲ್ಫ್ ಪಾಲಿನೇಟ್ ಮಾಡ್ತಾ ಇದ್ದಾನೆ. ಸೊ he has crossed this R with Y. So then he got R, Y. So then he has crossed this R with small y. Again sittu capital R, small y. Then he crossed this small r with capital Y. He got small r, capital Y. Then he crossed with small r with small y. He got small r, small y. So again ಆಯಿತು, ಈಗ he got the segregated alleles. ಸೊ ಸೆಗ್ರಿಗೇಟೆಡ್ ಅಲೀಲ್ಸ್ ಅಂದರೆ ಇಲ್ಲಿರುವಂಥ ಅಲೀಲ್ಸ್ ಏನು ಮಾಡ್ದ, ಸೆಗ್ರಿಗೇಟ್ ಮಾಡ್ದ. ಮೆಂಡಲ್ ಹ್ಯಾಸ್ ಸೆಗ್ರಿಗೇಟೆಡ್ ದ ಅಲೀಲ್ಸ್ ಟು ರೈಟ್ ದ ಚೆಕ್ಕರ್ ಬೋರ್ಡ್ ಫಾರ್ F2 generation. That means to self pollinate the next generation parents. Okay, so he has now segregated. These are the segregated alleles. So now by taking this segregated alleles, so what are the segregated alleles? We have capital R. So we have capital R, capital Y. We have capital R, small y. We have small r, capital Y, and we have small r, small y. So these are the segregated alleles. So ಇಲ್ಲಿ segregate ಆಗಿರುವಂಥ alleles ಅನ್ನ ಇಟ್ಕೊಂಡು ಏನು ಮಾಡ್ತಾರೆ, Mendel has self pollinated to receive the next generation's offsprings. So Mendel has self pollinated. Self pollinate ಅಂದ್ರೆ ಏನು? ಸೊ ಇಲ್ಲಿರುವಂಥ ನಮ್ಮ segregated alleles ಅನ್ನ, ಸೊ ಸೇಮ್ segregated ಅಂದರೆ ಅಲ್ಲಿರುವಂಥ alleles ಅನ್ನ ಅದರಿಂದಾನೇ ನಾವೇನು ಮಾಡ್ತಾ ಇದ್ದೀವಿ, ಕ್ರಾಸ್ ಮಾಡ್ತಾ ಇದ್ದೀವಿ. ಸೊ ಕ್ಯಾಪಿಟಲ್ ಆರ್, ಕ್ಯಾಪಿಟಲ್ ವೈ. ಸೊ ಕ್ಯಾಪಿಟಲ್ ಆರ್, ಸ್ಮಾಲ್ ವೈ, ಸ್ಮಾಲ್ ಆರ್, ಕ್ಯಾಪಿಟಲ್ ವೈ, ಸ್ಮಾಲ್ ಆರ್, ಸ್ಮಾಲ್ ವೈ. ನೋಡಿ ನಮಗೆ ಏನು ಸಿಕ್ಕಿದೆ ಇಲ್ಲಿ ಸೆಗ್ರಿಗೇಟೆಡ್ ಅಲೀಲ್ಸ್. ಸೊ ನಾವು ಸೆಲ್ಫ್ ಪಾಲಿನೇಟ್ ಮಾಡ್ತಾ ಇದ್ದೀವಿ. ಸೆಲ್ಫ್ ಪಾಲಿನೇಟ್ ಮಾಡ್ತಾ ಇರೋದು ಇನ್ ದ ಸೆನ್ಸ್ ವಿ ಆರ್ ಕ್ರಾಸಿಂಗ್, ದಟ್ ಈಸ್ ವಿ ಆರ್ ಕ್ರಾಸಿಂಗ್ ವಿತ್ ದ ಸೇಮ್ ಸೆಟ್ ಆಫ್ ಪೇರೆಂಟ್ಸ್. ಓಕೆ, ಇಟ್ಸ್ ವೆರಿ ಈಸಿ. ಸೊ ಹೇಗೆ ಸೆಗ್ರಿಗೇಟ್ ಮಾಡ್ತೀರಾ, ಇಟ್ಸ್ ವೆರಿ ಈಸಿ. ಸಿ ಯು ಆರ್ ಸೆಗ್ರಿಗೇಟಿಂಗ್ ಫ್ರಮ್ ದಿಸ್ ಟು ದಿಸ್, this to this, this to this, and this to this. So ಅದೇ ರೀತಿ ಪ್ರತಿಯೊಂದನ್ನು ಕೂಡ, and from here to here, here to here, here to here, here to here. So ಈ ರೀತಿ ನೀವೇನು ಮಾಡ್ತೀರಾ, ಕ್ರಾಸ್ ಮಾಡ್ತೀರಾ, ಪ್ರತಿಯೊಂದನ್ನು ಕ್ರಾಸ್ ಮಾಡ್ತೀರಾ. So when I cross this two bit, I get r by ry. So r y ಅಂತ ಬರ್ತಾ ಇರೋದನ್ನ ನೀವು ಹೇಗೆ ಬರೀತೀರಾ, ಸೊ r ಅನ್ನೋದನ್ನ ಒಂದು ಕಡೆ ಬರ್ಕೊಳ್ಳಿ, y ಅನ್ನೋದನ್ನ ಒಂದು ಕಡೆ ಬರ್ಕೊಳ್ಳಿ. ಸೊ r y. So here capital R, capital R, capital Y, small y. Then capital R, small r, capital Y, capital Y. Capital R, small r, capital Y, small y. So ನಾನು ಹೇಳಿದ್ದೀನಲ್ಲ, this to this, this to this, this to this, and this to this. Next, capital R, capital R, capital Y, small y. Capital R, small y, small y. Capital R, small r, capital Y, small y. ನೋಡಿ ಇಲ್ಲಿಂದ ಇಲ್ಲಿಗೆ, ಇಲ್ಲಿಂದ ಇಲ್ಲಿಗೆ, ಇಲ್ಲಿಂದ ಇಲ್ಲಿಗೆ, and finally ಇಲ್ಲಿಂದ ಇಲ್ಲಿಗೆ. Capital R, small y, ಸ್ಮಾಲ್ y. ನೆಕ್ಸ್ಟ್ ಇಲ್ಲಿಂದ ಇಲ್ಲಿಗೆ ಸೇಮ್ ಕ್ಯಾಪಿಟಲ್ R, ಸ್ಮಾಲ್ r, ಕ್ಯಾಪಿಟಲ್ Y, ಕ್ಯಾಪಿಟಲ್ Y, ಕ್ಯಾಪಿಟಲ್ R, ಸ್ಮಾಲ್ r, ಕ್ಯಾಪಿಟಲ್ Y, ಸ್ಮಾಲ್ y, ಸ್ಮಾಲ್ r, ಕ್ಯಾಪಿಟಲ್ Y, ಕ್ಯಾಪಿಟಲ್ Y, ಸ್ಮಾಲ್ r, capital Y, small y. Finally, we get here, see from here to here, here to here, here to here. See, right, capital R, capital Y, small y, capital R, small y, small y. So, I'm sorry, the last set I just did a okay. So just a minute, I am just finding a small, okay, fine. Next is small r, you get small r, then you get capital Y, and then you get small y. Finally, you are getting small r, and then you get small y, small y. So you get something like this, my dear students. You get something like this when you cross between these set of parents. Okay, now. So, ma'am, please upload PDF. Anta many are asking. Definitely, I will upload in the in the Telegram group. Yes, fine. So now these are the offsprings which we got in the F2 generation. So these are all the offsprings of F2 generation. So what do we get in F2 generation? ಏನು ಸಿಕ್ಕಿದೆ ನಮಗೆ ಅಂತ ನಾವು ಐಡೆಂಟಿಫೈ ಮಾಡಬೇಕು. Already I told you the phenotypic ratio, right? So let us now look at how many parent, that is offsprings, are round yellow, or how many different type of offsprings we got. See, ಏನಿದೆ capital R, capital Y, capital Y. Capital R ಅಂದ್ರೆ round, capital Y ಅಂದ್ರೆ yellow. So we got round yellow. So I'm just writing round yellow here. So what is this? This is also round yellow. So ಒಂದು capital R ಇದ್ರೆ ಅದು round, ಒಂದು capital Y ಇದ್ರೆ yellow. So because they are the dominant character. This is also round yellow. This is also round yellow. So this is also round yellow. This is round yellow ಇಲ್ಲ, small y ಇದೆ. So this is round yellow. So ಇಲ್ಲಿ round ಇದೆ, yellow ಇಲ್ಲ. So ಇಲ್ಲಿ round yellow. And ಇಲ್ಲಿ round yellow. ಇಲ್ಲಿ round ಅಲ್ಲ, wrinkled. So ಇದು round yellow. So we got round, no yellow. Round ಇಲ್ಲ, wrinkled. So how many totally we got round yellow? ಎಷ್ಟು round yellow ಸಿಕ್ಕುತ್ತೆ ನಮಗೆ totally? See 1, then we got 2, then we got 3, then we got 4, then we got 5, then we got 6, then we got 7, and then we got 8, and this is the 9th one. So how many round yellow seeds we got? 9 children. We got 9 round yellow. Now let us look at the next set. Next ಏನಿದೆ ಅಂತ ನೋಡೋಣ. Okay na? So next see here. So how many round green are there? Let us look at round green. Okay, see here this one. This is round. Okay. So let me highlight this. Okay. So what is this? This is round. Illirodu round and this is green. Okay, round green. This is round and green. Round green. Nodi green andre complete agi small y irbeku. And then see illi wrinkled ide, illi round green ide, illi wrinkled ide. So totally ಎಷ್ಟು ನಮಗೆ round green ಸಿಕ್ತು? 3 round green ಸಿಕ್ತು. 3 round green. So now let us look at the next. So ಇದೇನಾಗಿದೆ? wrinkled yellow. So we got wrinkled and we also are getting yellow. So let us look at how many wrinkled yellow. So wrinkled yellow. So wrinkled yellow, and here we got wrinkled yellow. How many wrinkled yellow we got? 3 wrinkled yellow. And last ಅಲ್ಲಿ ಇರುವಂತ ಈ ಒಂದೇ ಒಂದು organism, it is called as wrinkled green. So ಇದು wrinkled green ಆಗಿದೆ. So totally how many we got? Totally we got so 9 round yellow, 3 round green, 3 wrinkled yellow, and 1 wrinkled green. So this is the phenotypic ratio. What is this? This is the phenotypic ratio of the Mendel's dihybrid cross experiments of F2 generation. So ಇದೇನಾಗಿದೆ? F2 generation ನ phenotypic ratio ಆಗಿದೆ. ಯಾವ ಹೈಬ್ರಿಡ್ದು? ಮೆಂಡಲ್ನ ಡೈಹೈಬ್ರಿಡ್ದು. So this is the Mendel dihybrids. What is this? This is the phenotypic ratio. Always ನೀವು ಯಾವುದನ್ನೇ ಕ್ರಾಸ್ ಮಾಡಿ, ಡೈಹೈಬ್ರಿಡ್ ಆಗಿ ಯಾವುದನ್ನೇ ಕ್ರಾಸ್ ಮಾಡಿ F2 ಜನರೇಷನ್ ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ? 9:3:3:1 ಬರುತ್ತೆ. Okay, I hope you understood this concept, my dear students. So if you have any doubt, you can ask me in the comment area. So here are few interesting questions which I would like to discuss with you. Yes. The first question is, an angiosperm plant having red colored flowers when crossed with the others having the same colored produced 40 progenies, out of which 30 plants were with red colored flowers, 10 plants were with the white colored flowers ಅಂತ ಕೇಳ್ತಾ ಇದ್ದೀನಿ. So what I am telling, an angiosperm plant having the red colored flowers. So we always have to denote, when I say red color, it is capital R, capital R. You can take any letter. So for the color, so as I am taking capital R, so now this will be the dominant trait. Okay, when this crosses with that is this red color flower, when crossed with the other having the same colored progeny. Okay, so 40 progenies are produced. 40 progenies are produced. ಸೇಮ್ ಅಂತಂದ್ರೆ ಏನು? ಸೊ ಈಗ ನಾವು ಸ್ವಲ್ಪ ಥಿಂಕ್ ಮಾಡಬೇಕು. ಏನಾಗಿದೆ ಅಂದರೆ ಸೇಮ್ ಅಂತ ಹೇಳ್ತಾ ಇದ್ದಾರೆ. ಸೇಮ್ ಅಂತ ಹೇಳಿದ್ರೆ ಏನಾಗ್ತಾ ಇಲ್ಲ, ದಿಸ್ ವೋಂಟ್ ಬಿ ದ ಪೇರೆಂಟ್ಸ್ ಆಫ್ ದ F1 ಜನರೇಷನ್. F1 ಜನರೇಷನ್ ಪೇರೆಂಟ್ ಏನಾಗಿರ್ತಾರೆ? ಯಾವಾಗಲೂ ಒಬ್ರು ಡಾಮಿನೇಟ್ ಆಗಿದ್ರೆ ಒಬ್ರು ರಿಸೆಸಿವ್ ಆಗಿರ್ತಾರೆ ಅಲ್ವಾ? ಇದು ರೆಡ್ ಆಗಿದ್ರೆ ಇದೇನಾಗಿರ್ಬೇಕು, ಪಿಂಕ್ ಅಥವಾ ವೈಟ್ ಏನಾದರೂ ಆಗಿರಲಿ, ಸ್ಮಾಲ್ ಲೆಟರ್ನ ಕೋಡ್ ಮಾಡಿರಬೇಕು. ಸೊ ಅವಾಗ ಏನಾಗುತ್ತೆ, ಸೊ ದಿಸ್ ಇಸ್ ದ ಡಾಮಿನೆಂಟ್ ಟ್ರೇಟ್, ದಿಸ್ ವಿಲ್ ಬಿ ದ ರಿಸೆಸಿವ್ ಟ್ರೇಟ್. But they are asking something here, the red colored flowers when crossed with the other having the same colored, same andre enagutte, so that will be the self pollination. So andre now we have to look for the parent of F1 generation. So F1 generation na parent na we have to identify, because same anta heldaga we are always that we are always looking or we are always getting the same kind of parent only in the F1 generation. ಸೊ ಏನಾಗ್ತಿದೆ, F1 ಜನರೇಷನ್ನ ಆಫ್ಸ್ಪ್ರಿಂಗ್ ಏನಾಗಿದೆ, ಕ್ಯಾಪಿಟಲ್ ಆರ್ ಸ್ಮಾಲ್ ಆರ್ ಆದರೆ ಏನಾಗುತ್ತೆ, ಸೊ ದಿಸ್ ವಿಲ್ ಬಿ ನೌ ರೆಡ್ ಕಲರ್ಡ್ ಫ್ಲವರ್ಸ್. ಸೊ ದಿಸ್ ವಿಲ್ ಬಿ ದ ಪೇರೆಂಟ್ ಆಫ್ F2 ಜನರೇಷನ್. ದಿಸ್ ವಿಲ್ ಬಿ ದ ಪೇರೆಂಟ್ ಆಫ್ F2 ಜನರೇಷನ್. ರಿಮೆಂಬರ್, ಓಕೆ, ದಿಸ್ ವಿಲ್ ಬಿ ದ ಪೇರೆಂಟ್ ಆಫ್ ವಾಟ್? ದಿಸ್ ವಿಲ್ ಬಿ ದ ಪೇರೆಂಟ್ ಆಫ್ F2 ಜನರೇಷನ್. ಸೇಮ್ ಅಂತ ಹೇಳಿದಾಗ ವಿ ವಿಲ್ ಗೆಟ್ ದ ಸೇಮ್ ಕೈಂಡ್ ಆಫ್ ಪೇರೆಂಟ್ ಇನ್ ದ F1 ಜನರೇಷನ್. ಓಕೆ, ಸೊ ಇದನ್ನ ನಾವೇನ್ ಮಾಡ್ತಿದೀವಿ, self pollinate ಮಾಡ್ತೀವಿ. So when I cross now, that is when I self pollinate now these alleles, capital R, small r, capital R, small r, I get something like this. So capital R, capital R, capital R, small r, so capital R, small r, and I will get small r, small r. So I get the checkerboard like this. So ee ರೀತಿ ಸಿಗುತ್ತೆ. So now look at this. See, they got 40 progenies. Let us take this completely as 40. Let us think this as 10, this as 10, and this as 10, and this is also 10. So we got 40 progenies here. See, out of 40 progenies, if you observe out of 40 progenies, that means the offsprings, the 30 are the plant which are bearing the red color flower. So which are those? So this is bearing the red color flower because there is dominant trait. This is bearing the red color flower. This is having the dominant trait, and this is also bearing the red color flower. This is also having dominant trait. So how many total red flowers are there? 30 red flowers are there. Okay, and 10 plants are now with the white color flower. So F2 generation offspring nal kottidane namge white color flower bandide anta. That means ಏನಾಗಿದೆ, F1 generation parent ಯಾರಾಗಿದ್ರು ಇಲ್ಲಿ, so red has crossed with the white color flower. This is what we will call it as application based question, my dear students. Okay, so now you got white color flowers. Okay, so now out of 40 progenies, 30 are red and 10 are white color. Okay, so what is my question now? What is my question further? What is the possible genotype of the parent plants? What is the genotype? See, always I told you to remember to write genotype and phenotype of Mendel's monohybrid cross experiment. How do you write genotype? You look at the genes, that how many complete homozygous are there, how many complete heterozygous are there. Let us now write the plants we have got in the F2 generation. That is the offsprings we got in the F2 generation. Okay, so we got capital R, this one. Okay, then we got capital R, small r, then we got capital R, small r, then we got small r, small r. So this is completely homozygous, same type of allele. We got 1. This is completely heterozygous. We have both dominant and recessive trait. There are 2. This is completely homozygous because there are same type of alleles. So this is also 1. So homozygous red, heterozygous red, homozygous white. So this is the genotype, children. This is the genotype. So which trait is dominant here? Obviously you can identify which trait is dominant. Can anybody answer which trait is dominant here? So which is the dominant trait? Can anybody answer? Let me see who will answer fastly. Which trait is dominant here? Okay. Yes, Udaya has already answered. Red is the dominant trait because we got 3 red flowers. Okay, fine. So the next question is, what is the cross called? What is the cross called as? So what kind of cross is this? This cross is called as Mendel's Monohybrid Cross. What is this called as? This is Mendel's Monohybrid Cross. And what is the genotype? Genotype I have already wrote here. That is 1:2:1. So what will be the phenotype? The phenotype will be, obviously it is 3:1. 3:1 is the phenotype. Okay? Yes. I hope you got the answer, children. So ಒಂದು ಏನು ಟೆಕ್ನಿಕ್ ಅಂದರೆ ಇಲ್ಲಿ ಸ್ಟಾರ್ಟಿಂಗ್ ನಿಮಗೆ F1 ಜನರೇಷನ್ ಪೇರೆಂಟ್ಸ್ನ ಕೊಟ್ಟಿಲ್ಲ, ಡೈರೆಕ್ಟ್ ಆಗಿ ಏನು ಕೇಳ್ತಾ ಇದ್ದಾರೆ, ಸೇಮ್ ಸೆಟ್ ಆಫ್ ಪೇರೆಂಟ್ಸ್ ಆರ್ ಕ್ರಾಸ್ಡ್ ಅಂತ ಹೇಳ್ತಾ ಇದ್ದಾರೆ. ಸೇಮ್ ಸೆಟ್ ಆಫ್ ಪೇರೆಂಟ್ಸ್ ಯಾವಾಗ ಕ್ರಾಸ್ ಆಗ್ತಾರೆ? ಸಿ ದಿಸ್ ಇಸ್ ಸೇಮ್ ಸೆಟ್, ಕ್ಯಾಪಿಟಲ್ ಆರ್ ಸ್ಮಾಲರ್ ಅಂಡ್ ದಿಸ್ ಇಸ್ ಕ್ಯಾಪಿಟಲ್ ಆರ್ ಸ್ಮಾಲರ್. ಸೇಮ್ ಸೆಟ್ ಆಫ್ ಪೇರೆಂಟ್ಸ್ ಕ್ರಾಸ್ ಆಗೋದು F2 ಜನರೇಷನ್ ಅಲ್ಲಿ. ಓಕೆ, F1 ಪೇರೆಂಟು F2 ಜನರೇಷನ್ ಅಲ್ಲಿ ಸೇಮ್ ಆಗಿ ಕ್ರಾಸ್ ಆಗ್ತಾರೆ. ಸೊ ಯು ಮಸ್ಟ್ think a little, so you get answer very easily. I think you got the answer and you also understood the question. Let's go for next question. The next question is, a pea plant with the blue colored flower denoted by capital B is crossbred with a pea plant with the flower denoted by small w. So here completely alleles are changing. Let us take. So what kind of the plant they have taken capital B has crossed with the small w. What is the expected color of the flower in the F1 progeny anta kelta idare. A pea plant with the blue color flower denoted by capital B crossbred with the pea plant with the white flower denoted by small w. Okay, so little bit you have to think here. Why? Because here they have changed the the type of the alleles. They have changed the type of the alleles. So it is important to go through. So now we have, so just make this width as white. This is white color flower. Okay, so when we cross this, what happened? We get capital B. When we cross capital B, we get capital B, and here we get small letter w. So as they have already mentioned, children, they have already mentioned this is capital, this is small. So always when we cross between capital and small, what happens? What do you get? You get blue or white. Capital will be the dominant trait, right? So what kind of flowers we get here? Obviously we get blue color flower. Correct. So what is the expected color of the flower in F1 progeny? It will be obviously blue color because blue is the dominant. They have mentioned in the capital letter. What will be the percentage of plants bearing the white flowers in F2 generation? Let us take this parent. Okay, B and small w, capital B and small w. When you cross, we get capital B, small w, capital B, small w, and we get small w and small w. Obviously, what is my answer? I got the answer something like this. So what is my answer now for F2 generation? How many white flowers you got? You got only one set of white flowers. This is completely homozygous, the recessive character. Okay, that is white. Okay, how many you got? So one set of white flower we got in the F2 generation, because this is on self pollination we are getting. We are getting this on self pollination. Okay, on self pollination we got this. Next, state the expected ratio of genotype of capital B and capital B small w in F2 progeny. So what will be the genotype, children? Always, first of all, before writing genotype, just think whether it is monohybrid or dihybrid. This is monohybrid. Why? Because they have taken blue and white. They both are the traits of color, contrasting characters, but they both are related to color. So there is one particular character here, that is color. So monohybrid alli yavaglu genotype enagirutte? The genotype of monohybrid will be the 1:2:1. 1:2:1 is the genotype of Mendel's monohybrid cross experiment. Okay, Mendel's monohybrid cross experiments genotypic ratio is 1:2:1. Okay. So let me read now the last question of today's session. How do proteins control the characteristics of an organism? Okay, how do proteins control the characteristic of an organism? Students, see, we can see tall plants and also we can see the dwarf plants. We can see tall plants and also we can see the dwarf plants. See, we know that if the plant is growing tall, there is a hormone present in the plant and that is responsible for the growth of the plant. Even when the plant is not growing properly, if the plant is dwarf, that time also who is responsible? So the hormone is responsible, correct na? So a hormone proper ಆಗಿ ಸೆಕ್ರೆಟ್ ಆಗಬೇಕಂದ್ರೆ ಬಾಡಿನಲ್ಲಿ so efficiency of enzyme ಇರಬೇಕು ಅಂದ್ರೆ enzymes what is present in your body, that is now as we are talking about the plant, the enzymes present in the plant body has to work efficiently. So enzymes, when they work efficiently, what happens? It releases more amount of hormone. When they releases more amount of hormone, plants will be tall. So which is the hormone responsible for the growth of the plant? Let me just ask the question. So which hormone is responsible for the growth of plant? So name the hormone which is responsible for the growth of plant. Name the hormone which is responsible for the growth of plant. I'm waiting for the answer. You can write your answer in the chat section. Name the hormones responsible for the growth of the plant. So nobody has answered yet. Okay, fine. So let me tell you. So the hormone which is responsible for the growth of the plant is auxin, dear students, because auxin is synthesized at the shoot tips of the plant. This will help the plant's growth. If auxin has to release more, these enzymes which are present in the plant has to work efficiently. When auxin is released more, the plant will be tall. If the auxin is secreted less, it is released less, then the plant will be dwarf. So that is what the answer for this question. Because the proteins, where are proteins? Proteins are present in the DNA. Proteins are present in the DNA, deoxyribonucleic acid, right? So here, when the DNA releases more amount of, the hormone, when it prepare more amount of protein which is responsible for the auxin, definitely the plant will be tall, right? So the answer will be, we know that the plant hormones that can trigger the growth of the plant. Okay, so plant height is depend upon the amount of particular hormone released in the plant. If the amount of hormone made by the plant is more, then what happened? Then that is nothing but enzyme is working efficiently. Okay, so the amount of hormone made by the plant depend upon how efficiently the enzyme work. That means if enzymes are working efficiently, enzymes correct ಆಗಿ work ಆಗ್ತಿದ್ರೆ ಏನಾಗ್ತಾ ಇದೆ, ಹಾರ್ಮೋನ್ ಪ್ರಾಪರ್ ಆಗಿ ರಿಲೀಸ್ ಆಗುತ್ತೆ. So if the enzyme which is responsible for the growth hormone work more efficiently, then what happened? Hormones also releases in the more amount. Okay, so as a result of it, what happened? The plant will be growing tall. If the enzyme which is responsible for the growth of the hormone work with the less efficiently, then the hormone also releases less and the height of the plants also is less. So adrinda enagutte, genes control the characters of the plant. Where are their genes? The genes are present in the DNA, and DNA is the one which is helpful for releasing the proteins which is needed for the development or which is needed for performing all the tasks in our body. Ella tasks nu perform madake idene help madutte. Fine. So that's all for today's class, my dear students. I hope you understood all the concepts. So today we have learnt about what is meant by monohybrid cross, what is meant by dihybrid cross. So firstly we understood monohybrid cross, then I taught about dihybrid cross, then later on we understood what is meant by F1 generation and F2 generation. So followed by I have taught you how to write the Mendel's dihybrid cross experiment. Later we have discussed about the concept based on the monohybrid checkerboard. Okay, that is Mendel's monohybrid cross related 2 questions I had discussed. And finally, we have discussed that how the proteins control the characters of an organism. See, one thing you remember, it may be any kind of character which they give in the examination. So all the kind of characters are being exhibited by the hormones which are present in the plant body. But when we talk about the tall and dwarfness of a plant, that is being dependent, it is depending on a plant hormone which is depending on the growth of the plant. So the hormone which is depending on the growth of the plant here is the auxin. Auxin is responsible. Okay. So if auxin is released more, the height of the plant will be more. If auxin secreted will be less in the plant, then the height of the plant will be obviously less. Okay. So that is the one thing you have to know about the proteins. Okay. So I hope you found this entire session useful. If you find this video useful, don't forget to like the video, and after completion of the video, do mention your valuable comments in the comment area, my dear students. So in tomorrow's class, I will be coming up with the next concept of the lesson, heredity and evolution. Okay, until then, take care, study well, and have a nice day.